So let me tell you a bit about the work that we've been doing in Lausanne. I will. It will be really a mixed talk, like most of my talks are, where you're going to see also a lot of experimental validations of many of the designs we do, particularly on the protein design arena. But I think I would like to draw your attention, even before we start, to this aspect that I find, for me, that has become more central as we keep working on problems on protein engineering in our lab, about the importance of representation, right? So how do you actually represent an object so that you can capture the largest amount of information, and how can you actually modulate it in the most efficient way possible, right? So I think that will be really the common denominator of the few things that I will tell you here during my talk. So just as, let's see if this goes. Okay, this goes. A few items for the menu here. So I'll try to tell you about how we've been using the NovoDesign to explore new structural folds and what has been our approach. Also, a second topic of our work on how we're using surface fingerprints for protein function prediction and design, and lastly, some highlights about how we can use these surface patterns for Lagrangian discovery. So I think that, throughout these days, and even for those of you that don't really work so much with proteins, you are well aware of what proteins are. So they're absolutely magnificent molecules. They can do many, many different things. The information that encodes those proteins is stored in our DNA, which then gets translated into amino acids. And the 3D structures that are formed by these strings of amino acids is really what give us biological function. And clearly function and structure are related, but there's yet a lot of important challenges on how do we actually translate how do we actually translate structure into function. And so, but just to give you some examples here on the many different functions that proteins can have in biological systems, and this, of course, is by no means an extensive lecture, but clearly you can have defense mechanism, storage, transport, communication, structure, and even chemistry as catalysis. Now, the conversation for a protein designer always starts, or often starts, here. So one of the major challenges that we face, and again, this is common to many problems in molecular design and other problems that are now being tackled in very interesting forms with machine learning techniques, is that the number of possibilities is absolutely huge, much more than we can even conceive in our own brains. So, for instance, you can think about a protein which for all purposes and means, in a biological context of 100 amino acids, which is a very small protein, is not really that impressive. The number of possible combinations that you can have is way lower than the number of atoms that are estimated to exist in the universe. So often I like to think about this problem as we have sort of like this very, very large landscape of protein sequence space. And clearly, one of the things that we've been realizing as more and more organisms in nature have been sequenced, that the space of proteins that has been explored by natural organisms is much smaller than what it's actually possible. And it's sort of like clusters in islands, right? And I guess whenever nature gives us a clue for us to start engineering, oftentimes this is a more reasonable exercise. But when we want to sample outside of these islands that nature already gave us some glimpse into, this becomes a much harder problem. And so one of the ways to do this is to use computational methods. And so here again, just an illustration for you in case you don't think about protein design day and night. What happens here is that if you're a structure-based design person, oftentimes you start with an idea of what that structure could look like, or you essentially generate one. And then the challenge here is to find the combination of amino acids, which will result in a folded protein that you can then hopefully test experimentally. And the hope is that while we are in this process of designing new proteins, I think design is fun, but oftentimes we forget that while we are designing, it would also be interesting in that process to learn some of the fundamental design principles. And of course, we know some of them, but it seems like that these molecules still hold a lot of secrets. And at the same time, engineering proteins that can open up possibilities for new biotechnological applications. So typically the idea here is if we want to sample outside of the space that nature has already sampled, typically what we use is a group of techniques that we call, or that we often and colloquially refer as to the NovoDesign. And many different de novo design proteins have came into life, let's say it this way, in the last few years. A lot of that work actually comes from the lab of David Baker, and there's a few other labs that contribute also to this effort in a relatively regular basis. Now, one of the things that we have started to figure out is that it seems like it's a lot easier to design for structure rather than for function, which obviously clearly means that when we are simply encoding structure, we are missing some of the principles which are important for the biological function of these molecules. I should also just say that the word function in biology, it's a very complex word because for many different people means very different things. But let's just say that in this case, at minimum, besides folding, a protein should have some other kind of biochemical measurement, being that biochemical measurable, being that some kind of protein-protein interaction, protein-ligand interaction, catalysis, so some type of molecular activity that one can measure beyond folding. Okay, and so we've started, so again, reiterating this thought that oftentimes we can generate structurally accurate proteins, but it's much harder to encode function into this protein. So early on in our lab, we started thinking about how to actually solve or address parts of this problem. And basically what we came up with was this. This is still the good old times where we were using Rosetta Topo Builder or the Rosetta software suite, which basically we were doing Monte Carlo fragment insertions, which now this sounds a long time ago, but it actually wasn't that long ago. But the field has been moving very fast, and now this actually sounds like the past century. But maybe some of these principles can still help us to try to understand what are the needs that we actually have in order to try to encode for function in these proteins. And so what we did here was basically try to come up with an algorithm where we could define structural protein structures at different levels, particularly using 2D representations, then connecting these 2D representations and then translating these representations into 3D so that this could then be used in exercises like this, where we would extract a functional site out of a protein. So what you see here in green is not super important, but it's a viral protein. We took one of these sites that we knew that would be important for viral neutralization, and we started assembling a protein around this blue nugget, which we call the functional site. Once we have this assembled protein, then we start sampling across different configurations in order to stabilize these motifs, and ultimately we will drive sequences which were which could sustain this particular protein structure. Now, what we applied this in the past was to basically design vaccine antigens. And so just very quickly, we basically extract structural fragments from viral proteins. We use this Topo Builder algorithm and create small molecules, or small proteins in this case, which present the structural fragments of the viral proteins, immunize animal models, and study the antibody responses that were elicited by these antigens. So a few things happened here. It wasn't an astounding success as a vaccine design exercise, but we have learned a lot of the things that we could do in order to embed functional motifs into proteins using the NovoDesign approaches. Now, what we realized during this process was that, we couldn't really design super sophisticated proteins, and part of the reason is shown in this slide. Where, let me see if I can change this thing. And it's again related to to how we were representing proteins. So basically here, in order to create this novel proteins, what we were doing was basically writing strings, which we called forms that represent three-dimensional structure. We did not invent this notation. Somebody else, in this case, Willie Taylor, several years ago, more than a decade ago, has came up with this type of representation. And we found that it was a very interesting representation for us because it would allow us to work on the string space and then translate this into three-dimensional protein structures. But what we realized is that when we translated this to three-dimensional protein structures, and I will talk a lot more about this in the next slides, that these protein structures were not natural. And by not natural, what I mean is they didn't have the physical features that native proteins look like. And of course, that these physical features are, in a sense, imposed by the 20 building blocks that were used, that are the amino acids. And as such, our hope is that, okay, well, let's just start with a rough draft of what the protein looks like, and then we pipe this information into Rosetta, and Rosetta will fix it. Well, that didn't work out at all, because whenever you would put a template into Rosetta to basically create improved templates, the only thing that we could get out was basically templates that were at least as bad as what we would put in. And so we realized that what we had to do is, in fact, apply a number of geometric corrections just so that this three-dimensional representations of protein structures could, in fact, resemble more the physical constraints that natural proteins have, so that then this could help us in the search for the correct sequence. So the idea here is that if you have the wrong starting template and you use that to prime your sequence search, oftentimes you end up in places where the sequences are not relevant because your starting point is wrong. Now, we did this in a super clunky way, doing sort of like, database searches and finding sub-motifs within these structures and applying these transformations and then creating better models and then predicting the models. So this also worked to a certain extent, but it was super heavy and really troublesome to run. So what we started to think about is, well, okay, in principle, all that we're talking about here is how can we learn if these 3D sub-objects, in fact, what are their spatial orientations? And can we actually learn those patterns of distances and angles in between these secondary structures that you see here? And so this took us to this next generation algorithm that we created, which is much more learned based on features from protein backbones. And so the idea here is what we started using TR Rosetta networks, which were basically also developed in the Baker lab. And again, you can think about them as the predecessors of AlphaFold or methods on the same category of AlphaFold, albeit much less sophisticated in this thing. But basically, what was shown is that you could either use these networks to basically create designable backbones with target configurations, or basically by hallucination and just letting the algorithm creating new topologies. So what we really wanted to do here and was to basically jump from having backbones, which we call these naive sketches, which have non-natural features, into models that have much more natural features. And if you see here, is basically just some type of very small rearrangements in between the orientations of the secondary structure and some of the angles that compose the protein. Now, I will call your attention to this central panel here, mostly to again bring to the discussion the value of representation. So perhaps many of you are familiar on how a lot of these prediction problems represent protein structure. And one of the representations that is the most useful and that has been the most used are what we call contact maps or histograms in some situations. And so what you can see here is that, for instance, for this particular protein fold, you look at the histogram. So again, this is just a matrix where you have the contacts, the distances between the different residues in the sequence. And you can see that in many of the folded, or in this case, but this is true for other proteins, that there are very well established patterns of contacts in between the different secondary structures which compose these proteins, right? And so you can also have this type of representation for angles inside of the protein. But what you can see is that once you start distorting these structures, let's say, for instance, in the case of this sketch, most of these patterns are in fact lost, right? So you don't really see none of the patterns that are present on the native protein, and you can even have sort of like some kind of intermediate representations which are a little bit less distorted than the sketch. We call this the corrupt sketches. This is going to be important for the next slide, where some of the features are lost, but not all the features are lost. Okay, so what we thought about here is that, okay, so the challenge here is how to go from a contact map that looks like this, and can we actually sharpen it, if you will, into looking something like this? And can we train an algorithm that can do this for us in a very fast and efficient way? And so what we came up with was this genesis, where essentially what we did, or what we had to do, was to do a two-stage train, where we first use corrupted structures. And you can think about this as mildly distorted structures. We pre-train the encoder and the decoder in order to learn how to go from these corrupted structures into native structures. And then this was then used to essentially make a bigger jump, where we actually have sketches where the structures are completely blurred, if you will. And now we can actually transition from having these blurred features into protein structures that have much more distinct features. And so, and to give you an idea on how this overall pipeline looks like, now one can start from a sketch and basically enumerate many of the possible structures that nature has in fact never sampled, start from these blurry feature maps, create much more sharpened maps. These maps can then be posed or can then be input into some of the prediction tools that are available. In this case, we used a year ago or so, we used TR Rosetta, but this could also be compatible with other tools right now, and create these 3D structures. Now, the conversation is, what is important in this pipeline, because there's clearly a lot of steps happening, right? And so what you see here is basically a comparison of different pipelines, where we start piping just sketches and using this information with TR Rosetta and then a design step, or if you use the sketch plus Genesis plus TR Rosetta. So again, a little bit of an ablation study in terms of what the computational workflow looks like. And so clearly, if you just start from the sketch and the Genesis and do design, this will not work very well, as you see here. But as we start adding more layers of information, we can start seeing that we do better in terms of recovering the structures which we're aiming to have. And that's basically what you see on the y-axis, this TM score, which is basically a measurement of proximity towards the target structure. The higher, the better. And so you can start seeing that, for instance, and these are helical proteins only, strand proteins, strand-rich proteins, or helical and strand-rich proteins. So, and as you see, as the more sophisticated the process becomes, or the more information we get into this process, the higher is the recovery of the target structures that we want. So, and of course, this is just on the training set. On the test set, this is also true to a large extent, depending on the proteins that we're looking. The algorithm starts running in a lot more, or the network starts running in a lot more issues when it's to recover mixed structure proteins, meaning that it doesn't really generalize all that well, at least in this particular case that we have had. But still, since the results look at least mildly encouraging, our challenge was really this idea of, can we actually now design what has been defined as the dark matter of the protein fold space? And again, here I will call up the work from Willie Taylor, where essentially several years ago he has done this very beautiful work on trying to map the folds that nature has sampled more frequently, or that has sampled more rarely. And so, and using a bit this information, we could actually use this definition of sketches and creating these sketches of these protein folds that we're going to call the dark folds, and then try to design them using the Genesis pipeline. And so just to give you an idea, if in fact these folds are really that dark, we have here examples of 3 different folds that really proposed as being non-existent in nature. And in fact, we go and we check if there's any structure that is similar to that, both in the protein data bank or in the AlphaFold data bank. And some of the first structures that we encounter that are at the 6 angstrom root mean square deviations, which basically means that the first matches you encounter are not really the same fold of these dark folds, right? So then we pipe this through the Genesis and TR Rosetta pipeline. We create new sequences. We do what is now customary. Once we create new sequences, we pipe those sequences into alpha folds. And what you see coming out is that ultimately a lot of these sequences, in fact, have high TM scores towards our target sequences, so towards our target structures, as you can see here, very low root mean square deviations, and also high confidences, which is always a good thing. Now, this is again a computational dream. We have no idea if this can or cannot work. So we actually made a lot of these proteins in the lab, and so this is a very loaded slide, but ultimately there's a very simplistic message. So what you see here is when we actually made these proteins, we purify them and we see how they look in a size exclusion chromatography. So we just want to know if they're good biochemically stable and behave in solution. The mid experimental data that you see here is how well folded they are according circular dichroism. And here is an experiment where you see how stable they are according when you start heating up the solution. And what was surprising for us is that out of these 32 candidates that we actually characterized experimentally, at least 19 of them seemed promising in terms of having well-behaved proteins that were also folded and that could also be stable. So you also will see some proteins here that are not super well-behaved, and some of them which are also not extremely folded, as you can also see in some of them. So we have a mix of many things, but it was an interesting success rate for the type of exercise that we were doing here. And again, this is still work ongoing, and we're still trying to get structures out of many of these proteins and characterize many more designs. So the conclusions, or a few thoughts for this part, is that we could, by imposing these native topological features into these backbones, we could improve the results to have success rates, good success rates in the sequence outputs, and that this idea of sharpening topological features can be learned and is a fast strategy to bring these native-like features into the novel proteins. This Genesis sharpening process also allowed us to go beyond native folds, and we could create what we like to call this dark folds. Okay, so let me start a new conversation now, which again the common denominator is related to protein representation. And so in here, I will tell you about the work that we've been doing in collaboration with Michael Bronstein for a few years now, which is about studying protein surfaces. So again, as many of you are aware, proteins can be represented in many different ways, particularly if you're trying to represent a protein structure. You can either represent the secondary structure or individual atoms, or you can have more of a near continuous representation, which basically represents the water, the interface with solution, where typically you map the solvent excluded volume of this molecular object. Now, what is interesting about this particular type of representation is that you can actually study it, or you can actually map properties into it, which oftentimes you don't do at the atomic level. And particularly, I will speak about geometric properties, which are often not very used in atomic representations. But you can also map into it chemical features. And so you'll see a few on how we actually dealt with all this information in the next few slides. So the idea here, and the question that started us up for this work was really if there was some type of surface patterns which could help us to understand and ultimately engineer functional features of proteins. And the parallelism here is, if there's these patterns that you can learn from pictures, let's say from human faces, are there the same patterns that we can learn in protein structures that then will determine their function? Now, the way that we implemented this was by creating this computer program that we called Massive, which ultimately what it is a framework that generates numerical descriptors that encode the features of the surface of these proteins. And so here you can see one of these protein structures represented as its surface. And one of the first things that we do is to basically describe or define these geodesic patches where there are points inside of these geodesic patches, which then are enriched with geometric features of their local environment. And so the geometric features that we're talking about here is shape index and distance-dependent curvature. So perhaps many of you are aware, but clearly one of the properties that is fairly common in proteins, in particular for molecular recognition events, is that whenever there's, for instance, a protein interaction, typically one of the necessary requirements is that these proteins are not only shape complementary, but they're also chemically complementary. And then in this case, you can see that for the chemical features, we started mapping different types of features into these surfaces. Together with this, we set up a system of polar coordinates, just so that each point inside of the patch could understand what was its location relative to all the others, and then applied a geometric deep learning architecture, which would allow us to basically generate these fingerprint descriptors, which are nothing more than numerical values that aggregate all this information and can be trained for particularly different applications in protein modeling. And three of the applications that we initially explored was basically this idea: given a protein pocket, can we identify the ligand that binds to that pocket? Given a protein structure, can we identify which sites are more likely to interact with other proteins? And then, as a logical follow-up, we also used this surface fingerprints to guide docking exercises. And I won't go too much into the details of each one of these applications, perhaps just highlighting the docking aspect of it, because it's going to be relevant for the last part of the talk. But basically what we saw is that we could learn very efficiently which surface patches from different partners were actually compatible with each other and likely to make a protein-protein interaction. And so basically this is what you see here, that we could train Massive very efficiently to describe those or to identify those that were interacting patches versus non-interaction patches. And here we actually applied a trick which is relatively simple, but it worked very efficiently, where we trained a network with the capability of minimizing the distance between the patches and the descriptors of the patches of the interacting ones, and maximizing those that were non-interacting patches. Now, two results came out of this exercise. One of them was that we were pretty much just as good or a little bit less good than the best docking algorithms around, but there was one major difference. We could be we could be more or less at the same level of performance, but much, much faster. And so, and this was important for the next topic that I want to tell you. Now, during this process, there has been a number of different improvements in Massive. Again, our initial version was probably one of the ugliest pieces of software that you can imagine, and it had many external dependencies. Ultimately, a brilliant graduate student in a lab, Freyer, was able to basically make just an end-to-end architecture, which was not dependent on many of these external softwares. And now things work a bit more smoothly. And ultimately, we were able, even using the initial version of our Massive software, to basically tackle a different problem, which I've been interested for a few years now, which is this idea of designing de novo proteins to target other proteins of interest. Again, you can imagine that this sounds like a simple problem, and I think, to a certain extent it should be a simple problem. However, there has been many efforts, and this really remains an important task, which for now we don't have a robust solution. And part of the issue there, it's likely related to also an incomplete representation of the objects that we are working with, because a lot of the proteins all the proteins that we are working with, or the way that we're represented, are basically represented as static snapshots. And as we know, molecules are not static snapshots. There's also other aspects that in biological systems and in solution, right? We have all this contribution from water. And here in many of these situations, we also don't account for that type of importance and contributions from solvents. So a number of important issues which many of our methodologies don't really know how to deal with. So in the end, we have a very limited representation of the molecular objects, because again, we represent them as static molecules, which they're not. And then we still try to engineer molecular recognition events, in this case, protein-protein interactions, which clearly are dependent on a number of very small interactions, right? So very few residues and atoms, if you think about it, are determinant for making a protein-protein interaction occur versus non-occurring. Now, protein-protein interactions are super important, right? So they're basically, you could probably think about them as the basis of how biology works, right? In order for function to happen, different molecules have to interact with one another. Of course, it's not the only thing that is happening in biology, but it's definitely one of the most prevalent molecular recognition events. Now, so how are we going to use Massive now to design the novo PPIs, right? And the idea here was basically, okay, let's start with some target protein. So here it's not super important what it is, but in this case, we used PDL1, and we drive a surface fingerprint from the site where we want to that we want to bind the design protein. We create also a fragment database, and in this case, the reason why the speed comes in a very important why the speed is very important is because we were able to create very large fragment databases, in this case in the order of the 100 million, and still be able to scan through all these possible binding candidates very, very quickly. And that, I think, it has been a differentiator of the approach that we've been having. And ultimately, once you do this scanning, we are then able to score many of these interactions. We get candidate interaction motifs, and once we have this candidate interaction motifs, we can then place them into different proteins and see if they in fact bind to the target protein or not. And so now we're going to start more heavily on the experimental data. But to show you a couple of examples where we did this, so we took again one of the proteins that has been in fashion these days, which is the SARS-CoV-2 protein, and the idea here is, can we actually design a binder which can inhibit the attachment to the host cells? And so we started checking which kind of sites will be more targetable. So that's one of the differentiating aspects on how we tackle this problem. The first step of our pipeline is really to just get the target protein and ask to the massive site network which sites are more targetable, and then focus our efforts in designing interactions for those sites. So we don't go randomly across all the other sites in the protein, but rather we have very focused efforts in terms of targeting the sites. And so then again, we place segments or fragments, if you will, and we create different designs. And in this case, we created 63 different designs, and then we screen them using yeast display. It's not super important what it is, but let's just say that in this case, we can actually screen many designs without having to make each protein individual. And so what we got from this first screen was that, in fact, one of the designs did show a very minor binding interaction, and we'll get into this. And by minor, I mean low affinity. So the interaction between these two proteins was not very strong. So we started with with the DBR301, which was our original computational design. And then we started putting mutations on top of it in order to improve its affinity. So what you can see here is that ultimately, once we had one protein that we could measure, we were at the level of the micromolar range of affinity. But with a few mutations, we could in fact lower this affinity, or improve the affinity into 80 nanomolar. So a few more functional characterizations, basically showing that this protein was able to, in fact, inhibit or neutralize the virus. But I would like to now go to another example and tell you also how we dealt with this particular design problem and what we have learned from it also in order to make the process more efficient. So here what we're going to be doing is to try to target PDL1. So PDL1 happens to be one of the flattest interfaces or surfaces that is present on the PDB. And oftentimes, if the surface that you're trying to target is flat, that means that you don't have a lot of features that you can establish protein-protein interactions, or it becomes relatively hard. And so this is the site, again, as predicted by massive, that we could actually target. So what we did here was basically, to run again this search for protein interaction motifs. We get a wide variety of matches, but eventually there's one where the algorithm seems to be more confident, or it's more often seen as a protein-protein interaction motif. We then analyze if there is some type of if there is some type of sequence convergence in this motif, and we start seeing that in particular positions, in fact, there's always a subset of amino acids that arise from this clustering algorithm. So we select one of these seeds, design 5 scaffolds, ordered 16 designs, and then we try them experimentally. So again, the results that I'm showing you here are for experimental binding, and the take-home message is really this idea that typically we get binding from the computer, but this is very residual binding, very low affinity binding, to the point that we have to go and mutate the protein on some key residues to figure out if the binding is actually specific or not. And so you see here an image of one design, an image of a different design, again with the same type of biochemical behavior. And then what we try to do is actually to run one of these high throughput experiments where, well, the question is really this. Well, so if we design these things in the computer and we can't achieve high affinity straight out of the computer, what is going wrong? And this experiment is basically designed to tell you this, right? So basically what we do in these experiments is we mutate all the residues of the protein by all the remaining 19 amino acids. And then we can actually learn, so if you would change for a particular residue, which was not the one of your original design, do you improve, or do you get worse in terms of binding affinity? And so the results are actually very interesting, because they basically tell you that, yes, you can improve if you change residues that are outside of the binding seed that it was predicted by massive. So meaning that some of the residues that are also important for the protein-protein interaction are not optimal. But typically, the residues that we selected with a surface fingerprints were pretty much close to optimal, which was interesting for us to see. Now, again, we measure this affinities. We can improve these molecules by doing mutagenesis, basically from the high micromolar level into mid nanomolar level. So that's already pretty acceptable affinities. But what is important is, of course, can we actually check if our predictions are right or wrong? And so what we did here was to basically solve crystal structures of some of these molecules together with our collaborators. And so you see here the model that was predicted and the crystal that we obtained, and they're basically pretty much spot on. So indeed, things here were working, or these protein-protein interactions were being established in the right modes of interaction. Okay, so, but maybe one of the questions that you might be having is, well, are these protein-protein interactions in fact really novel? Because particularly in the case of PDL1, we know which one is the native binder. The native binder is called PD1. And what you see is that PD1 in fact utilizes a very different type of motif, so basically a very different type of solution in order to engage the same site. But when we actually engage it, we engage it with a helical motif. And so basically showing that even using elements that nature has not used yet, we can in fact create new protein-protein interactions. Now, what we would like is mostly to run a computational algorithm and obtain a bunch of binders straight out of the computer without having to be optimizing them using in vitro evolution and mutagenesis. And so what we did here and what we started noticing was that in principle, it was necessary to optimize areas of the protein-protein interaction before we actually obtain binding straight out of the computer, right? And so what we've learned from here is that many of the designs that we had had in fact very poor hydrogen bonding interactions across the interface. And so by doing a few steps of design, particularly refining the seeds or some elements of the seeds and refining the scaffolds in order to enrich these interfaces with hydrogen bonds, we could then try to obtain that. And that's what we see here. So ultimately here, what we did was, okay, we ordered 2,000 designs. We again screened them, and straight out of the computer, we could already have clones here, which were binding into the protein of interest. And here, just some pieces of data. So I'm going to go a bit faster over the 2 slides, maybe just to show you some other usage of the surface fingerprints. Let's see. So let me just try to close this part here. And so ultimately, what I've showed you here is that these vector fingerprints do reveal these functional signatures from protein structures, and they have enabled this ultra-fast docking that we can now use for the design of site-specific binders with high accuracy. Clearly, we still have a number of important challenges, even on how can we actually use this information for instance, for more native generative models, if you will, and to basically optimize the surface descriptors even for other types of tasks. And now, just as the last 3 or 4 minutes that I think I still have available, I hope it is, Max. If I'm going too long, please just cut me off. But so just a few slides over on how we can also use these surface fingerprints, for instance, for tasks related to ligand discovery, which I find super exciting because it really means that in a way, this representation is able to capture a lot of the principles that are guiding molecular recognition, independently of these are, if these are from small molecules or proteins. And so part of our adventure here started with this idea: can we actually identify fragments that could bind to particular pockets? So I already told you a bit about that at the beginning, but so here the idea is, and maybe this slide will be a bit better. So the task is really, so if we have a pocket, a protein pocket, which we represent in this schematic point clouds, and if I extract subsections of this pocket, am I able to predict which kind of chemical moiety is able to establish a productive binding interaction? And so that's what we set out to do. And essentially, in one of our first attempts, we could already be relatively close, depending on how we actually rank these matches compared to also other algorithms, which in this case were not based on machine learning approaches. But we could already be in relatively compatible performance levels. Again, I think the important aspect of this task is that for many of, or for those of you that have tried to do small molecule ligand docking, it is a very messy problem. And so here really our idea was, if we could explore a different representation of the protein target that would allow us to extract more reliable information, can we actually improve, even if marginally, this process of docking small molecules or very small molecules, small moieties of these small molecules into these protein pockets. And so giving you some examples as well, some examples in some test cases that we had in the lab by taking the ligand, showing you what the real fragments are. So by hiding or masking part of the ligand, can we actually recover what the fragments look like? And in many cases, we do recover fragments that are in the space, in the chemical space for which the native ligands have. So that was good. But now, of course, the next question is, well, so if you can place this fragment so well in the pocket, what are you going to do with this, right? And clearly the question is, well, we'll have to do some kind of fragment linking. And this is when Max came into the picture, and clearly, we have this super exciting phone calls that we discuss about everything and anything. And one of the at the time when we started this, Max was also super excited about this diffusion models, and that's how we got into it with his help. And the challenge here is really, if we have a bunch of disconnected chemical moieties that are now placed into a pocket, so this would be in the concept of a pocket, although the pocket is not really shown here. Can we reconstruct the ligands in a reliable way? And so, to do that, Ilya, a student in the lab, trained the diffusion model, which I'm sure many of you are already very familiar. But here the point was, there's a few tasks that one needs to do. Essentially, you'll have to be able to predict the probability of what the linker size that you want to build. And once you have had this probability, you can then start diffusing the moieties to connect the different chemical parts by this process. And here, just showing you some examples on how, in fact, the process looks like. Ultimately, we also benchmarked this against other algorithms that could do the same. The metrics, particularly, some of these metrics for me, I'm still a bit not super comfortable with them because they're essentially just computational assessments on how drug-likeness these molecules look like or how synthesizable it is. In general, the metrics look okay. I think, I'm by heart also an experimentalist, so I really only believe things when experimental tell you that they're good. And but in general, it seems like the trend that came out of there is that we were, in fact, building molecules that obey to many of the chemical rules. It's still debatable if many of them, for instance, would be synthesizable, and that's a problem that we're actually looking into. But we could have algorithms that create many valid molecules, and that would also be unique and create some kind of chemical diversity. And of course, one of the things that we were really excited about doing is to condition this process into the pockets of the targets, which many of the algorithms for molecule generation in machine learning actually don't do. And for us, this was the exciting part. And what we could see here is that, in fact, conditioning this to the pocket really makes a difference in terms of reducing the number of atomic clashes that you observe. And of course, this would be hopefully what you would expect, but the hope here is that we can translate this into focusing the chemical search into more useful region of the chemical space. Okay, and so this is basically for linker design, and just to give you an example on full molecule design. So no linker, now you go and you just present a pocket and you create a full molecule. That was the next extension that was done by Arne. And the only thing that I want to show you here is this one result showing you that when you generate the ligands, ultimately these ligands are also, according to other orthogonal methods, in line with the native ligands in terms of their predicted binding energies or affinities, as you will, and some of the root mean square deviations that are generated by our diffusion model are also recovered with lower MSTs. So with it, I think that we're just starting. So a lot of this work is very new in our lab, and there's really a lot of things to work on. So it's unclear to me yet right now what's really the optimal representation for many of these molecular objects that we're working. So it seems like we can already extrapolate and create new molecules. Again, in the space of small molecules, we have not validated any of these experimentally, but in the space of proteins, we've been able to validate things to a very detailed extent. And so, of course, I think that, the future hopefully will be very exciting, and that we will be able even to further extend the applications of these surface fingerprints to other molecular design tasks. Okay, so with this, the most important slide, which is to give a big thank to the team in Lausanne and all the hard work that they have put in many of these difficult problems that we work, which oftentimes there's a lot more failures than successes, and so I have to really thank them for their enthusiasm. And thank you all as well for your attention, and I hope that I didn't take too much time, and I'm happy to take any questions.